Good morning, everyone. Hope you're well and welcome to today's webinar organized by the Eastern Branch of the Institute of Food Science Technology, IFST, on food structure, appetite, and energy intake. This webinar is sponsored by Foods, which is an open access journal by MDPI. So just a couple of housekeeping points. So throughout the webinar, if you have any comments or questions, feel free to put them on the Q&A box, which you'll find below at the end of your Zoom box. After the talk that Professor Well will give, we'll have a chance to go over the questions. We'll try and respond to as many as we can. Okay, so for those of you who don't know me, I'm your host for today. I'm Dr. Federico Bernuzzi. I'm a postdoc over at the Quadram Institute of Biosciences. I look at how food bioactives affect our microbiome. And today, Professor P. Wild, who is a research leader here at Quadram, will give a talk about his current research. Professor P. Wild is a group leader on food structure and digestion at the Quadram Institute. After graduating in a degree in biophysics from UEA or the University of East Anglia, the same year began his scientific career at the Institute of Food Research on the spectroscopy of proteins. He's now a group leader focusing on understanding how the microstructure of food changes during the digestion and how that impacts our health. So with that, I'll hand over to you, Pete.
So I want to talk to you today a bit about the role of food structure on appetite and energy intake, and it's based on some work that we've been doing for a while, and hopefully give you the take-home message around that it's not just about the composition of the food that we eat, but also about the structure, and specifically around sort of nutrients, bioavailability, and energy intake and appetite. So it'll be a mix of sort of background around the subject, and also some examples of our research and other people's research. So starting off, really starting at the beginning around how I believe that food structure has influenced our own evolutionary development, and our early ancestors, once they started to transform the food that they would gather or hunt by cooking or milling or grinding, and that transformed those raw materials into something that's much more digestible, it's much more energy dense in terms of the assimilation that our body can make of the energy and the nutrients, and usually more delicious as well. An example here is a classic example where you can see the amount of starch that has been hydrolyzed within potatoes, and from raw potato starch, raw starch is not digestible. But as soon as you process it, you cook it, you mash it, you make it into crisps, whatever, all that starch, or the vast majority of that starch, becomes available, and you instantly transform the nutrient content of that food without changing the actual composition, purely by cooking it. And many people think that drove some of our evolutionary development. Certainly the coincidental development of our brain size a couple of million years ago coincided with these developments in our ability to cook and transform food. And also preservation as well allowed us to preserve foods to last for longer, to keep us through seasonal variations. It improved our ability to capture and utilize energy and nutrients, which improved health and reduced foodborne diseases of our ancestors. And a lot of people believe this underpinned our physiological development and our global population expansion. And so moving into a bit more detail, as I said, our early ancestors, they ate foods which were structurally rich, unprocessed, And the nutrients are very difficult to extract, very slow to digest. And so we developed a digestive system which was able to extract as much nutrition as possible from these highly structured foods. And you got this very prolonged release of energy, great seasonality. And this is all controlled by our appetite signaling networks, controlling our satiety to control how much food we take in. As I said, as we further developed our ability to process and transform those foods, this went in parallel with our evolutionary development. So what are the upshot for modern man? Well, most people think that this has gone a little bit too far, and that our overconsumption of heavily processed foods, these foods are structurally poor. They've been broken down, extracted, reformulated, and put back together again. The nutrients are easy to extract and digest. The nutrients and energies are absorbed rapidly. And together with changing our eating patterns and eating on the go, convenience foods, and so on. It's a high glycaemic index, it's a low poor satiety index, and this has led to sort of the development of metabolic disorders. But the interesting thing is that this change in our diet, this transformation in our dietary patterns has changed relatively recently compared to our evolutionary history. So these are massive changes in very short space of time compared to our evolutionary pathway. So we have not evolved or adapted quickly enough to suit our modern diet. And this has several issues, and one of these is our obesity levels. As we all know, these are increasing over recent years. Data for England here, some historical data. Clearly, with different years, this is increasing. But also as we age, we get fatter as we get older, and this is a consistent pattern. And the reason for this is that our evolutionary history, we've evolved to consume more energy than we actually need. And our appetite and satiety signaling processes and pathways actually encourage us to eat more than we actually need, and store that excess energy as fat for lean times, for winter, for times when we don't know when our next meal is coming from. But the main issue with modern man is that we do know exactly where our next meal is coming from, and global international food companies, massive processing, preservation, as producing sort of low cost, high available foods, a constant all year round supply of food, of convenient food, easy to cook, and generally quite energy dense. And this has led to this gradual surplus of energy intake over a life course, lead to this increase in generally in weight gain over our life course. It's the classic middle-age spread, basically. Together with changing lifestyles, increasing sedentary levels, has led to increasing levels of obesity, type 2 diabetes, etc. And this is the rise. We know the rising costs of diabetes and obesity, but this is a global phenomenon now, and the data shows that for low and middle income countries, the levels of diabetes has surpassed those of many high income countries. So it's not just limited to developed countries. So going through our digestive system in brief detail, and really to give you a bit of background about why food structure is so important. So quickly going through the different stages of digestion. First, you consume the food, you break it up in your mouth. Amylase starts to break down some of the starch. This passes into the stomach, where there are more enzymes, and the stomach is a holding tank for our food, and it started to break down. There are some enzymes, some mixing, and acid pH, and then the food is gradually emptied out of the stomach into the intestine, where the main site of digestion and absorption takes place. And the structure of our food is important here because only small particles and low viscosity fluids are emptied out of the stomach because they are prevented by the pylorus, and large particles, highly viscous components, get retropulsed back into the main body where it's continued to break down. And this can influence how quickly the stomach empties into the small intestine where the main digestion takes place, with pancreatic enzymes, bile salts helping to break down the fat, and all the transport and uptake processes. And then finally, at the end of digestion, the remaining material is delivered to the colon, where it's fermented by our microbiota, producing various other beneficial metabolites that influence our health. But this is all controlled by a really complex feedback and signaling mechanism network. And this is as a gross simplification of that. And the signaling starts in the mouth with our sensory processes telling the gut and the brain what it's expecting. The stomach senses fullness, controls hunger, and gives feedback signaling to the brain and other parts of the gut. In the small intestine, it's mainly based on nutrient signaling, and that signals to the stomach, to the brain, various other organs, controlling gut motility and appetite and the rate of gastric emptying. And at the end of the small intestine, these signals become more intense. So if we're detecting valuable nutrients towards the end of the small intestine, then these signaling pathways slow down gut motility to try and maximize the absorption of those really valuable nutrients. So we don't want to lose too many of those nutrients to the colon. But it is important to deliver fermentable material to the colon, where things like dietary fiber are fermented, and again add to the signaling for gut motility and appetite control and so on. So the main upshot of this is the absorption kinetics, not the actual composition, is important to actually promote this complex feedback and signaling throughout the gut, which actually controls our digestion and appetite. And hopefully that'll become apparent as we go through this webinar. So what I mean by food structure, well, this could form a whole range of length scales. So it goes from the molecular level, how those individual molecules are assembled into larger structures, into organelles. The cell is very important because it can encapsulate all those nutrients within a cell and within a larger tissue structure, and ultimately to the level of the whole food itself, which has to be mechanically broken down. And all these length scales can influence the digestibility of those nutrients and their bioavailability. And I'll give sort of some examples as we go through. Sort of first of all, really on the molecular level, one example being glucose. So glucose is our most important primary unit of energy for metabolism. But we don't actually consume glucose in its monomeric form largely. It's mainly consumed in the form of a polymer of glucose, which are then broken down and digested in the gut. And this can take shape of a range of molecules, but this is just to demonstrate that these are all glucose polymers, but their molecular form actually influences their nutritional impact. So most of the glucose we consume is in the form of starch, which is the storage organelle in plants, and that can be in the form of amylopectin, which when cooked gelatinizes and forms this really open structure, which is very digestible and provides much of the glucose in our diet. The other form of starch is non-branched, and it's a linear molecule which forms helical and crystalline structures, which are very slowly digested, or they can be resistant to digestion with our intrinsic enzymes, but they are fermentable. So once they reach the colon, they're fermented by our microbiota, producing some beneficial metabolites which can aid metabolism. The animal version of starch is glycogen, which is again is a heavily branched glucose polymer, and that is readily digestible. And then the non-digestible forms of glucose include soluble fibers like beta-glucan, which again is a linear polymer with some branching, but the molecular links between the glucose polymers, individual monomers, render it indigestible by our intrinsic enzymes, but it's fermentable by the microbiota because of the cocktail of enzymes that they possess. And on the extreme side is cellulose, which is a highly structured crystalline form of glucose polymers in a three-dimensional network, which are definitely undigestible, but only fermentable only a very limited level in the human gut. But they require extensive fermentation, such as with ruminants who successively ferment the cellulose. So although these are all primary glucose polymers, their nutritional value is dependent on the type of bonds between the individual glucose units, the degree of branching, what sorts of structures it forms, and how that's affected by processing and cooking. And one example, which I'll come to shortly, is you can manipulate the amount of amylopectin and amylose in the plant by changing the activity of the starch branching enzyme, so you can create more amylose by not creating these branched structures found in amylopectin, and that can reduce the glucose levels in your bloodstream and increase the fermentation, so increases the amount of resistant starch. I'll come back to that with one of the research examples. So in terms of the importance of food structure on our health, there are quite a few examples of these, but the one I'd like to give is this one, which compares fruit and fruit juice. And there's been a whole load of studies published coming out of the healthcare workers longitudinal cohort study in the States, where around 200,000 healthcare workers from the US from 3 different cohorts have been followed up for about 20 years. Initially, they were all free of major chronic diseases at the baseline. And this data has been analysed and produced a number of papers. And this one really was focused on comparing the impact of whole fruit consumption versus fruit juice consumption of the same fruit. So they were comparing like with like. And interesting, they found that the greater consumption of specific whole fruits was associated with a lower risk of type 2 diabetes, whereas a greater consumption of the fruit juice of the same fruit was associated with a higher risk. And so the composition is the same, although there can be differences. You can, depending on the production of the juice, you could filter out a lot of the fiber. You're breaking down the cellular and tissue structure. And so there's a whole load of free sugars in there, which are readily absorbed. Plus the volume is very different. The rate of energy intake is different. So you drink a glass of fruit juice very quickly compared to eating an orange. Also, a glass of orange juice may contain the juice maybe from 3 or more oranges, which you would never eat in one sitting. So there are several reasons underpinning this observation, but most of those are around about the structural differences between the 2 types of food matrix and what impact that has on our consumption. There's a really good video by Kees de Cruyff where he shows one of his PhD students either drinking a litre of fruit juice made from a kilo of grapes, or eating a kilo of whole grapes. And the differences in the rate of consumption and the feelings of fullness were quite amazing. I can always send a link to that if people are interested. So, as I said, this eating kinetics is really important. This is a study by Kevin Hall's group, and they compared the two groups of people. It was a crossover study, so both groups did both diets, comparing an ultra-processed diet with an unprocessed diet. These individuals, it's free living, ad libitum consumption, so they could eat what they like, but they only had these either this diet or this diet to choose from. And if you compare the two diets, so this is the processed diet, and this is the unprocessed diet, both the same calorific content, both nutritionally same nutritional composition, but the energy density is completely different. The amount of food you need to get this amount of energy is very much less than the unprocessed food. There's lots of fruit, vegetables, and so on. And eating the processed diets actually led to an increased energy intake of about 500 kilocalories, which led over the 2 weeks to significant weight gain compared to the unprocessed food, which had a lower energy intake, and actually most people actually lost weight. Because the amount of volume you need to eat to get to the same sort of calorific intake is much greater, leads to greater satiety and appetite suppression because of the volume of the food you need to eat and the low energy density, and the fact that it takes you longer to eat these types of food, all contribute to these differences in energy intake. So, you know, this comes from a lecture by Kees de Graaf, as I said. Look at, there's a PDF version, and he's got a YouTube presentation as well if you're interested in this. They're really good talks. I'm interested in sort of the range of how food process can influence health in this way, but I'm also aware that we also need to look at how we can process foods to improve the health impacts of traditional foods, for example, to optimize the impact on satiety and appetite. So an example of the research that we did in this area was we had a project with the John Innes Centre and Imperial College, and it's largely focused on peas and resistant starch in peas, and it was focused on the Mendel's wrinkle pea. So this is Mendel's original, one of the mutations he studied between the round pea and the wrinkled pea, and here as I mentioned in a previous slide, the starch branching enzyme is knocked out. So the wrinkled pea produces more amylose than the wild type, and there's clearly differences in the morphology of the pea, and the starch granules also look very different because these are much more crystalline structures compared to the native starch granule in the wild type pea. And we were interested in how this affected glucose absorption and metabolism. And the human studies performed by Imperial showed that there were differences between how well processed they were. So there were differences in the little r, the mutant pea showed lower glucose and insulin, and the whole peas showed a much bigger effect in reducing the glucose uptake and the insulin levels. And these were comparing two isogenic peas, which were their genetic background was identical apart from this one enzyme which is affecting the starch branching. So we're not comparing peas with wheat, for example. These are two identical products. So to see such a big difference was actually quite remarkable. And we found that not only was it around the starch, and going back to the example where you process the potato to make the starch more digestible, we found that with some of the peas, it was actually the other way around. It's a bit complicated, this figure, but basically, as you would expect, in the wild type pea, when it's cooked, you reduce the amount of resistant starch. But actually, in the mutant pea, the little r pea, when you cook and digest the peas, the amount of resistant starch actually increases. So this is something to do with the behavior of the starch, but also something to do with the larger structure. And this is backed up by measures of crystallinity using X-ray diffraction. So again, the increase in crystallinity, which is unusual, observed after cooking with these wild type peas, compared to the decrease in crystallinity that you would normally observe for most other things. And then looking at the larger structure, we found that we get a greater breakdown of the tissue structure in the wild type pea during and following cooking. So you can see a lot of breakdown here, a lot of cloudiness in the cooking water, which nutrients have been released, compared to the little r pea, which is, they all seem to be pretty intact. The mutant peas were tougher. They were stronger when you compress them, and they break down in a different way, and there were a lot more larger particles following chewing compared to the wild type pea, which released a lot of the smaller particles, which were individual cells or starch granules, which were then much more available for digestion. And we know, and this is the work of a colleague at Quadrum, that the particle size is really important for releasing nutrients. And so large particles will encapsulate and hold on to more of the nutrients within the cells, whereas smaller particles will release more of those nutrients relatively and encapsulate less inside those smaller tissue particles. And so for the same particle size, as you increase the particle size, the digestibility or the release of that starch becomes less. And as the little rp had more of the large particles and less, and the wild type had more of the smaller particles, then this helps to explain the differences in the in between observed in the human studies. So it was a combination not only of the changes in the molecular structure of the starch, but also how the larger tissue behaved during processing and consumption. Another example is dietary fiber structure. Everybody knows, and the evidence is overwhelming in terms of the impact of dietary fiber on health, but the mechanisms aren't always obvious, and they're not always well known. And we know that soluble dietary fiber is thought to modulate fat digestion and reduce cholesterol. The viscosity is thought to be important, but as I say, the mechanisms aren't really well known, and the human studies are really quite inconsistent in terms of the impact on human health. But the matrix structure of the food that is fed during these human studies could influence that. And we find that, for example, beta-glucan, a well-known soluble fiber, if it's in the form of oat bran, which is a mixture of insoluble and soluble fiber material, a crude form of fiber, you get really good binding of bile salts, retention of bile salts. Whereas purified beta-glucan has a very weak interaction with the bile salts and no significant effect. And if you just looked at the insoluble fiber alone, again, that doesn't have an effect either. So there seems to be some sort of synergy between the soluble and the insoluble fiber and the complexity of the matrix that is presented. And what we found is that oat bran fibers, oat bran is the particles as they are hydrated or digested, they swell and they form this layer of beta glucan around the cellular tissue particles, which are capable of retarding digestion. And we also found that they can stick together, form these larger aggregates which could entrap nutrients, stop or reduce the diffusion of enzymes into the matrix. And this was much more effective at binding bile and slowing down lipid digestion compared to soluble fiber, which has just been released or added to the food. So again, the matrix properties of even dietary fiber can have a big influence on the digestibility and nutrient release. And then also we've done some work on mycoprotein, which is the basic ingredient for corn. And mycoprotein is interesting because it contains intact mycelial cells, intact filamentous fungal cells, which form these fibers, which give the meat-like texture in these products. And human studies have shown that the protein is highly available. And we show that, and this is under the microscope, you can see these cell structures, these hyphal structures containing nutrients before digestion, and then they release their nutrients after digestion. And most of the action happens in the small intestine, and it's sensitive to different enzymes. So even though the nutrients are trapped inside those cells, we've shown that the enzymes can actually get into those cells. They're much more porous than plant cells and release that protein. And that structure we found also can reduce the glycaemic response. And we showed that the presence of increasing amounts of mycoprotein actually inhibit starch hydrolysis and inhibit the enzyme in the meal, in the digester. And we found that the enzyme amylase, which breaks down starch, actually diffused inside the fungal cell walls and was trapped there, and so it couldn't actually get to the starch which was in the continuum, and actually reduce the overall activity of the digester. And we also showed that similar effects with fat digestion, and this seems to explain why mycoprotein can have positive effects on glucose absorption, glucose levels, and fat digestion, and also is able to reduce appetite by slowing down the kinetics of digestion and nutrient release. Finally, I'm running out of time, and I mentioned earlier about sensory signals. And so, is a low calorie always the best option? And foods with a reduced energy content, but by mimicking the taste and texture of the original foods to make these healthier products, has been an aim of food companies for many years. And this is very good for people who are on a calorie controlled diet, who are determined to reduce their energy intake. But one has to be careful about what impact this might have. And the reasoning is that when we consume food, those sensory cues, whether they're sweet or thick or creamy, influences our perception, and we have this sort of expected satiety or expected nutrient intake. Whereas when you actually consume the food and you actually your body senses those nutrients, and we actually absorb those nutrients, and we get an actual nutrient status. And if there's a mismatch between what we expect and what we actually absorb, then that can influence our appetite response. And a study from Martin Yeoman's group at Sussex showed this a while ago, and we followed up with this with him in emulsion systems. But basically, what he's saying is that feeding people a preloaded beverage, whether it's either with low sensory, so it's thin and watery, or this is thick and creamy and sort of energy replaces, or it contains sugar and fat. And if there's a mismatch between the actual energy content and the sensory content perception of those foods, then you can lead to a big difference in the subsequent energy intake. So they drank this beverage, and after a while they had lunch, and they could eat as much as they liked. And basically, if you were fed a low energy but high sensory beverage, then you consumed more at that lunch. So this overcompensation from this reduction in energy is down to this mismatch between the sensory properties and the actual nutrient content. So whilst it's good to reduce energy levels in foods, a big mismatch between the sensory and the nutrient composition can have unintended consequences. So bringing this to the end, to summarize, hope I've got this message across that the food structure and its behavior during digestion has a huge impact on our health, in addition to the actual nutrient composition. Foods containing structures or structures which are resistant to breakdown, they generally are more slowly consumed, they are digested more slowly, the nutrients are difficult to extract, so they are released more slowly and on a more sustained level. This leads to a promotion of satiety. It can provide material for food for the microbiota. So really you need to consider the structure of the food as well as its composition. And I apologize for this next bit profusely. So you can have your cake, but you probably won't want to eat it. And on that note, I just wanted to thank you for your attention, members of my group, past and present, and collaborators for the sort of work that's been presented. Thank you.
Okay, thanks a lot, Pete, for this very interesting talk. So question and answer session now. There's really a couple of questions in the box, so I'll get straight into it. So the first question is, do you have any data on wheat flour particle size and whether it can affect the glycemic response?
I don't have that data, but some people have published data. A colleague of mine, Katrina Edwards, has looked at wheat and also legumes, and it seems to be a common issue with particle size and reducing digestibility. So the large particles seem to slow down the release of nutrients and release the accessibility of enzymes. I believe, though, don't quote me on this, that certainly in the case of pasta, the semolina flour in pasta has a large particle size, and this in part might explain the slower release of starch from pasta.
Okay, thank you. Next question. What is next with regards to your research?
Oh gosh. I think that the fiber work, I think, is really interesting. There's this link between the matrix structure of the fiber and really understanding how it interacts with other components of a meal during digestion. We really need to sort of secure those mechanisms, I think. There's a link at the moment, but they're in separate studies. So there are human studies which So we need to show some real human studies where we can manipulate the structure of the fiber in a way without changing its composition, and then show sort of beneficial effects. So then foods can be designed with fiber that's in the right sort of form, the right sort of matrix that has a real impact.
Okay, thank you, Pete. There's a comment in the box saying by one of the participants, very interesting, it makes me think about food in a different way. And then the next question is, so when we might see nutrition labels that reflect the true nutritional impact of foods rather than simple composition?
Oh yeah, that's from Peter, yes. Yeah, it's a really interesting question, and there has been a debate about this. I was at a meeting well, before lockdown, and that there was this discussion, and people are proposing different ways. So whether it's measured on like a glycaemic index, so or whether it's like a nutritional sort of impact, and it was quite a lively debate actually. And there's two degrees to this. You know, one is yes, it'd be great to provide that information to the consumer, but then the other side is that the consumer is already overwhelmed with information, and certainly the consumers who you know are in need of this sort of information are unlikely to be digesting, if you like, that additional information and understand what impact it has on their health. So yeah, it's a really interesting question, and there is an active debate around this. But there are certainly pros and cons with trying to provide even more information on foods.
What is your view on protein supplements as dietary aids?
Oh, that's an interesting one. Well, you know, I have to hold my hands up. I'm not a nutritionist, but protein supplementation. Yeah, there's a view that we already, certainly in developed countries, eat too much protein. But I know, you know, certainly for certain conditions, well, not conditions, but you know, for performance athletes require a lot of protein for recovery and muscle synthesis. There's certainly a lot of research into the elderly population and reducing the impact of sarcopenia. And the important thing there is whether you're having rapidly digested or slowly digested protein, and what impact that has on muscle synthesis and reducing muscle loss as we age. And I think that's a really important aspect of protein supplementation. But for normal healthy individuals, I don't think there's much advantage unless you are targeting some specific condition.
Do you have any examples on how the food industry can apply this research? Is there any opportunity for reformulating foods?
Yeah, I mean, there's lots of opportunities. The big problem with satiety over recent years is that it's been largely to do with EFSA, in that EFSA didn't regard satiety as a health claim, as a health benefit, unless you can actually prove that there is a causal link between satiety and appetite regulation and weight gain, or the development of type 2 diabetes or some other ill health. So getting health claims or saying on a packet that, you know, this increases appetite, or, you know, reduces appetite or whatever, is challenging. But there's certainly, from the food companies that we talk to, there's certainly a keenness to develop healthier foods where appetite is you know, can be a factor, and that certainly some consumers are demanding or wanting foods which will help them control energy intake. And I know that, you know, I gave the example of the mycoprotein. There's been lots of studies on mycoprotein and how it affects appetite and glycemic response. And so, you know, structuring those foods in a way to maximize those effects, I think, are going on. But the big issue, the big hurdle is legislation and regulation, how you can tell the customer, the consumer, what impact this food is having in a legal and you know, allowable way, I think is the big barrier.
I read there was a connection between gut bacteria and them being linked to us craving sweet foods. The more we eat, the more we want. Do you agree with this?
I don't know. I'm not an expert in that area. I know there are certainly there are sort of reward centers of the brain which respond directly to sensory signals, and that drives eating behavior. But I don't know anything about the microbiota link with that, I'm afraid, sorry.
What do you think are the drivers for why some food companies produce/market ultra-processed foods and not those with more nutrient density structure? How can these be managed?
Yeah, that's a really good question, Carol. It's a tricky one, and you know, ultra-processed foods have been sort of demonized recently, and there's a whole range of studies you know, showing their detrimental impact on health. But this is quite a crude tool, and there are ultra-processed foods which are clearly bad for us because of the nutrient composition and the energy density. But there are also those which are being designed to be healthful and control these effects. So I think we have to be sort of very careful about that. But primarily it's been cost, I think, and you know, the food companies, they're trying to sell a product. They're, you know, it's a capitalist venture, and it's not like the NHS, not like a healthcare system. It's driven by profit, and so the companies have to make foods which people want to eat, which actually will repeat buy, and they need to maximize their profit, and the impact of which is the sort of the foods that we see on the market shelves now, how that's evolved. But there are some companies who want to change that, who want to move towards you know, utilizing the sort of processing technologies available to produce healthier foods, certainly targeted at people who want to change their lifestyle, want to have a positive health message. So I think, like all systems, there are those who want to provide a positive impact on health, and those who are driven purely by profit.
And next question actually follows up on what you just mentioned. How would you modify processed food to retain its social benefits and consumer attraction, but tackle the challenges that it has in our digestion?
Yeah, that's really good. Another really good question, but it's really complicated, I think, because there are a whole range of consumer groups. I mean, the big retailers are really good at classifying people into different boxes, and some consumers will respond to information, advice, and adapt their diets and so on, and they don't mind changing their dietary patterns. Whereas other people, they want foods which taste good, give them pleasure, they're cheap, convenient, and they use those, and health is further down the priority list for those. So I think there's a different set of solutions depending on the consumer groups that you're trying to target. And certainly for the demographic that we are sort of interested in, we're looking at how we can manipulate in very small, subtle ways the structure of everyday foods, such as, you know, white bread, pastries, whatever, and regulate the digestibility of those foods so that consumers don't really notice they still buy those foods. And we've got to do it in a way that doesn't affect the cost too much. And that's a real challenge.
Have studies looked at the makeup of a meal as opposed to a single food or product? Might a solution lie in a plate or diet model?
Absolutely. And not something that we've looked at, but certainly there are groups around who look at the synergies between different foods in the same meal. Certainly when we looked at the microprotein, we looked at the extrinsic components. So it's not just about the microprotein itself, it's how it affected the digestibility of starch and fat. So, to have the effect, you need to consume these foods together in a mixed meal. So yeah, that's a really important aspect. And it's also the psychological aspect as well. There's a lot of work going on in, I think, in Bristol, around whole meal effects, but how you perceive the calorific energy content of a meal and how that affects your appetite signaling. So there's some really interesting sort of work going on, not only around how it behaves during digestion, but how psychologically you perceive those meals and how that affects your behavior. And things like memory, you learn which meals have high energy or make you feel full or taste delicious, and you learn those are learned behaviors which you adapt to. So there's some, it's way outside of my comfort zone, but there's some really interesting stuff out there about the whole meal scenario.
And we're also diverting a little bit more into sustainability now. Is there any research around if processed or unprocessed food better in terms of reducing energy carbon consumption?
I think there's a broad this is really complicated. Yes, I mean you can take on one aspect there's the drive away from animal production and animal protein consumption to more sustainable practices, and the food industry is in a position where, using the economics of scale, they can produce foods which minimize the amount of animal material and maximize the amount of plant material, but then provided in a form which is acceptable to the consumer. We've seen a whole range of plant-based burgers on the market, and they can be done either individually at home, but that's quite difficult. So you need the food processing industry to be able to do those at scale. But then also, if you think about if people go down to individual, almost like a sustainable lifestyle where you're growing your own fruit and vegetables and everything, that's not sustainable really, because certainly in very densely populated areas, larger cities in low and middle income countries, there's just no space to do that. So those people have to consume foods which are being produced by the food industry. So it's tackling that problem to make sure that the foods that are produced and made available to those people are healthy and having a positive health impact, but also are sustainable and address these issues of carbon production. So it's a massive question, Carol, that one. So I'm not going to do that justice in the next few seconds, but it is really interesting aspect of the whole equation of the food processing industry.
Okay, so we don't have any later. We don't have any more questions in the chat box, so I'll quickly ask you a question. So you had a lot of questions already that you answered. I was really interested in the slide that you showed, the mutant P, the small RR, that when you increased it, actually the resistance starch increased. I think you probably mentioned it already in one of the previous talks, but I kind of forgotten. Do you know what that is? Is it to do with the genetics or the P?
We have a hypothesis. But we sort of need a bit more sort of funding or more targeted research to prove it. One thing that we think is that the starch granule is a mixture of amylose and aminopectin, and for some reason, when the starch granule in the mutant pea is heated, a lot of that amylose comes out of the starch granule and appears to coat the starch granule with amylose. So when that is cooled or after cooking, it crystallizes around. So it effectively encapsulates the starch granule with an indigestible resistant starch. Whereas that doesn't seem to happen as much with the wild type. So that's why I think that the cooking, but also the crystallinity increases. So you're disrupting the starch granule structure, and then it's recrystallizing in a slightly different form, which increases the amount of crystalline starch and reduces the digestibility. So yeah, it's a really interesting transition. And it's been seen in some foods. I mean, there's the classic thing with pasta. You reheat pasta and you cool it down, and whether it's the type of wheat that's used in pasta, I'm not sure, but certainly there's something around how the starch recrystallizes in a different sort of structure that affects the subsequent digestibility of these cooked products. And if we knew and understand that, then we can, you can start designing foods by very simple manipulations, but they could have a massive impact on glycemic response.
Okay, we just had, I think this will probably be the last question. So we just had a question come in. Do you know if the flavor of the pea is affected with the two variants?
That's a really good one, because actually these peas were sort of really popular because a lot of the peas we're looking at were not garden peas. They are mature peas that are used as ingredients. So they're the mushy pea once they've gone to maturity. Garden peas are picked at immaturity before there's too much starch buildup, and the mutant pea actually produces more sugar. There's more free sugar around, so they're actually sweeter. And a lot of these peas are used as the sort of the garden peas that you get with your frozen garden peas, and they're selected because of their improved sweetness. So they do actually taste quite nice. But I'm not sure about the more mature pea. I don't think there's much difference in the taste of those. There's certainly the reports back from the human studies didn't show any difference. The main problem with peas is they taste like peas, so incorporating those into other products can be a bit challenging. But there are companies who are working with ways to control that through processing to actually reduce the pea-ness of the ingredients.
Okay, so we are reaching the end of the webinar. So thanks again, Pete, for your time, and thanks for all the participants who have joined today. If anyone still has additional questions, feel free to get in touch. I don't know what's the best thing to probably send an email to the digital IFST, and then they'll probably forward the emails to us. And then Pete, I'm sure Pete will be able to answer more questions.
Yeah.
I forgot to say that this session has been recorded, so it will be uploaded onto the IFST website, especially under the webinars region. I think that's all. I'd like to thank again for Foods for sponsoring this webinar. Yeah, and that's all from my side. So yeah, thanks again for everyone who attended this morning webinar. Wishing you all the best. Have a great rest of your day. Thank you. Bye.
Thanks, Fred. Thanks, everybody.